Welcome, everybody, and thank you for joining on Eagles Sunday. Like I was telling Don, I apologize for making Don compete against the Eagles, but I think the Eagles are going to win, so we all should be okay. Chuck gives me the thumbs up on that one. I guess he's watching. So tonight, uh, many of us know Don Coates. Don is a member of the club. You've been beekeeping, what, 15 years, Don, about?
Maybe 20. Yeah.
Maybe 20 now, longer
Yeah.
than I have. And he's been a member of the CCBA and has really uh, helped educate myself and many of our CCBA members about disease in the hive. He's a retired veterinarian, so he clearly has the science background to understand this type of work, and he really does a good job in presenting the material in a way that's practical for the backyard beekeeper. When I first started beekeeping, there was so much to learn, and disease sort of fell off for me. And I really can say I didn't know much about disease and hives except for mites for the first year or two or three. And then I met Don, and Don really presented in a way as like I start to understand it and understand that disease is in every hive. And I think that's going to be uh, some of what he presents tonight when he is going to present is how sick is normal. So thank you, Don, for agreeing to do this tonight.
It's been a good experience to review this. And it's actually an old PowerPoint I had when I had given the presentation to either EAS or the other programs. So it was fun to explore this. And as I mentioned a while ago, that we could maybe sometime or consider having a group review. Uh, my lab is functional. And I've got extra microscopes, and the club still has a microscope. Is that right, George?
We do.
So if anybody's interested, they can in doing this kind of activities, say meeting once a month or um, have an agenda to develop on a presentation. Um, and right away could invite even from what I have to present tonight, to invite any thoughts of members who have dead outs that they would like to have diagnosed. We can start that in a conversation. So we can go ahead with the main theme here. How sick is normal? And the fact being that there are uh, I labeled it as stressors are always present, but there are also, I mean, more specifically, there are pathogens always there as well. And it's kind of mysterious how they're otherwise kept in control. European foul brood or of course American, it'd be terrible. But I've had a case of the chalk brood and the the subtle ones, the viral infections, they are there too, and it's kind of mysterious how they do do manifest themselves. A quick sharing of another experience. I've had a hive that had a bunch of crawlers out front. This was in February, a couple of years ago, and the they were just going up on leaves of grass and hanging there. And it turned out to be a paralysis virus. Excuse me. And going through that memory reminds me too that members should know that there there are uh, labs to accept samples uh, that we can contact one of these labs and have maybe a working relationship to monitor diseases. Um, but there are special little boxes that you can purchase to send live bees from your hive to a diagnostic lab. Um, so we could pursue that. Um, Now getting back to the outline here, that they have bacterial, infections, fungal infections. Again, chalk brood, the common one, but there's also one called stone brood. It's apparently pretty rare. And the viral infections, which are are really latent. They're commonly there, but don't manifest very much. And besides the parasitic of the varroa mite, there's a condition called amoebic dysentery. Um, Randy Oliver has a, a branch subject on that on his website. And of course, should mention him that scientific beekeeping, um, I assume everybody's heard of scientific beekeeping, and his website, just a huge source of good information. Um, a factor that determines our functionality of the hive is whether we have a good queen or not. Um, these are, again, the list of stress experiences that the hive has within it. Um, Nutrition is a factor, as with us. A, um, a monoculture of forage is not good. Um, that is the broad spectrum of amino acids, um, even if they're going out and collecting pollen from the almond trees, as some of our packages that we buy are sourced from, uh, that they've been in California and the almond groves. Um, you kind of are concerned about how healthy they are coming to you with. They're not balanced because of the monoculture of their forage. It was all almond pollen. Weather cycles, of course, uh, have an influence on their health. It varies. Um, there's a factor of summer dearth that maybe some of the new beekeepers are not aware of, that I think some of our observers here will agree that a great percentage of our honey comes from trees. That the spring blooming of the trees, uh, one tulip poplar tree would be equal to perhaps 3 or 4 acres of wildflowers. And when they stop blooming, these different trees, which is usually like late May, going into the second week in June, why we have the dearth. It's the kind of the scraps of the forage. Um, and then the other factors of stressors on the hive, on the colony, is the mites that spread viruses, and even worse still, when there's an overwhelming infestation of the mites, it's the parasitic mite syndrome that will just decimate a colony. And we beekeepers are part of the issue. We can make mistakes in our planning, and maybe not like now. If you've overlooked insulating the top and perhaps even the sides of your hives, why you're adding quite a bit of stress uh, to your colony. Pesticides are always out there, um, and I forget the name of it, but there's a pest neonicotinoids are the residual pesticide in corn and soybeans. Uh, so be aware that if your hives are near an ag field of corn, particularly, why the neonicotinoids um, and maybe some similar chemicals from golf courses uh, are adding quite a bit of stress to your colony. This is a commercial logo that shows the components of health and function of your colony, the center being the bee, um, the genetics here of how strong the queen is. Um, and for us, well, this critter could be either the beekeeper not doing his management well enough. Um, he adds to the possible mistakes. Um, and again, the monoculture of a single bloom, not providing enough spectrum of amino acids. So there are different components, and I think somebody else who created this logo could probably tell more. Here's the spectrum of viruses. The uh, the deformed wing, I suppose people can see my cursor. We see the deformed wing. Yeah. Um, that's the more common one. And of course, that's spread by the varroa mite. Um, but there are others too. Most of them do not manifest any signs. But here's the chronic bee paralysis virus and acute bee paralysis virus. And again, these tend to not show any detriment to the hive. But here, Bee and Farm Partnership, which was an organization that in the past collected samples from volunteer beekeepers and have this background of information on what viruses and organisms are in your colony. Um, and here was the Black Queen uh, virus uh, is prominent, but it really doesn't cause much of a clinical sign. But the deformed wing virus does. So this graph doesn't uh, indicate the severity of the effect on the hive, on the colony. But they're out there. And for those who have not seen it, here's the bee with the deformed wing virus. Um, and of course, it's non-functional, can't fly, and um, you wonder how it gets nursed and, and fed. The poor thing is doomed. Um, but the communal nature of the behavior of honeybees, they'll probably feed her, keep her going. Another concept here is that we share.
John, there's a quick question before we go on.
Okay,
Vidge is asking what the basis of the test of what was the basis of the test on of the previous slide?
basis of the test of this one, or
No, the one, yeah, that slide. What was the basis of the test of the previous slide?
um, mechanically, you have to send in live bees to uh, University of Maryland was the function center for having them tested. That was in collaboration with the bee informed paralysis.
Did they do a DNA test, she's asking?
An A test?
A DNA test to do I assume they're viral PCR samples to
Yes, right,
determine the viral load. It
right.
looks like they're red, yellow, and green, I assume is viral load as far as viral count, I assume. It looks like the green was low viral count, the yellow is medium, and the red is high. So I think it's just showing that, deformed wing virus in 1,492 colonies was present in 50% of the colonies. And, black cell queen virus was present in 75% of the colonies. I don't know exactly uh, if this was just this seemed like just surveillance. I don't recall if this was just beekeepers just sending in samples or paid samples or what it was, but it looks like a just a screening of 1,492 colonies showing that uh, virus. I believe there probably PCR levels were quite prevalent and
Yes.
high in a lot of the samples. Sorry, Don. I'm
No, no,
blabbering.
that's wonderful. Thank you for the technical input. And it's a very old sheet or report. You can see that it was in 2013. And so the program is not underway anymore, but it would be interesting to touch base with them. And again, here's the lady with the deformed wings. Um, and a little comment of conscience that our honeybees are sharing the viruses with the native bees as well. And that transmission takes place out in the forages for the native bees, the bumblebee or the leafcutter bees or the various ones that have their special names. Um, there's said to be 200 different species of native bees in our area. Again, kind of re-emphasizing our concern for nature. Uh, our hobby has become a very popular and growing abundance of honeybees in our forage areas increases the risk of spreading diseases to native bees. Our chickens are loose and outnumber the natives and may be spreading avian flu. Here for resources of similar intent on diagnostics, um, the variety of print material is there. Would comment that this one right in the center is really antiquated, but I think it's been updated somewhat. A lot of my pictures come from this one, also produced by BIP, Bee and Farm Partnership. Um, this is a newer publication, and I found very helpful. I didn't get it till about a week ago, um, but it's for those interested in this pursuit. It's very helpful. It's got 28 professional contributors, um, and it's fairly new. It's got some old information, but a lot of new information as well. So I would certainly recommend that. Sidebar here, I guess the fact of nutrition is crucial for honeybee colony growth. And again, as mentioned before, um, the abundance of variety of pollen for them. The pollen, of course, is essential for the protein that makes the chitin of the honeybee. Uh, Once their exoskeleton is produced, that's it for the rest of their lifetime. Um, and the more robust and strong it is with a variety of protein materials, why probably the longer their lifespan and functionality would be. And then the bee diseases. I put these little printouts because they're notice that there's the icon of the video that they can be um, viewed by this website. Um, it's an agricultural website and by the company that publishes this book. So if anybody is wanting to take a picture of that and with their cell phone or get onto it, why that would be another good resource. So the cluster of diseases And let's see, yeah, these are, yeah, they're not all bacteria. Um, we got chalk brood, American foul brood, the bad one, European foul brood, which is manageable. The key being here that it's non-spore forming, but even though it doesn't produce a spore, which is the indelible nature of the spores in your hive boxes that spread from season to season. Uh, that's the bad component of American foul brood, and that's why when it's diagnosed, um, the the protocol is to burn them. It's a sad experience. Um, and then sac brood is viral. Stone brood uh, is, of course, a fungus, and chalk brood was a fungus. And the parasitic mite syndrome um, is the overwhelming infestation from mites. European foul brood. Now we'll have a few slides for quick diagnosis visually. Um, the organism of European foul brood sort of dissolves or eats out the substance of or the muscle protein of the larvae and leaves only the trachea. That's why you see these streaks. The substance of the larva has been digested, so to speak, or dissolved, and leaves only the shell of these tracheal tubes of the larva that are building because, of course, the honeybee does not have blood per se. It's got hemolymph in it. And all of the respiration takes place with trachea that go through the body. And these are the streaks of these trachea that remain because I guess they're tougher than the other muscle components of the body of the larvae. Um, again, this is European foul brood, not American, that it doesn't have that sticky cell wall on the outer frames. Um, it's it's a more gentle disease, almost so to speak. But um, the uh, the casts we'll see later too are stuck on the side of the cell wall, which you can see on the side view of a frame when you're trying to diagnose the after effect of European foul brood. They go through a stage of drying and desiccation after they're dead, and um, the remnants are casts, which would be like this, and the scale up on the side. When you look at the frame, the comb on at a side angle, you can see that's what caused your problem. Now, here's American foul brood. Uh, rather perfect example of how terrible the capped brood is. It's described as wet or sweating here where the little bubbles of the dissolved larvae are oozing through. And if you take a toothpick or a stick of straw and touching that, it makes that telltale sticky slime that's with American foul brood. So the stressors on, again, your hive is always on the threshold of decline. That is the stressors are the dearth in the summer, um, summer seasons that when we don't have good forage, bad weather, inability to forage period. Um, Small colony populations, if it's hard to get them to build up if they start small. Um, I guess what comes to mind right away when you buy a package, you have the option of getting a 3 pound or a 4 pound, and I would always go for the 4 pound to get enough enough population to get some momentum going. And we beekeepers make our mistakes as part of the stressor components. Again, quick review here. Uh, these this picture was of one of my hives that I had, I guess, neglected. At any rate, it got chalk brood in it. And you can see the what's termed the salt and pepper effect. Uh, this shows also a little tip I would comment. This little bib I put in front of my hives, um, particularly showing here was the chalk brood carcasses, but you get other tips too of a variety of indicators of what's going in the hive, what's being thrown out, how much uh, pallbearer activity there is, that they're carrying out dead ones, accumulating. So you get an indication of some trends in your hive by having this in front of the hive. Um, I have also just used a bit of cloth and pinned it down in front of the hive to catch the detritus that's there. Again, here's the chalk brood on the bib. Overemphasis of chalk brood is not that bad, but here we go with a virus now. There's the sac brood, um, the shrunken pupa, looks like a slipper. Not as decimating as some other diseases, but it's one you don't like to have there. And parasitic mite syndrome, um, comment here, I think I've got it in another slide also, but the drone brood, the bulging brood, um, are good indicators of mites or show you that your infestation, because the bulge gives more space for the mite to reproduce. Um, and there you see two of them in one hive, I mean one cell. So making an assessment when you're doing an inspection, be encouraged by uniform cap brood surfaces. That is, no perforations, no bulging except for the drone cells. You have a balanced, adequate workforce. The nurse bees that are keeping the brood area warm. Absence of patchy brood, which is the reverse of patchy brood is a reverse of uniform cap brood. Note too that I guess now we're still in a broodless period. Would you all agree that they're not producing brood and won't till what, end of February in our area?
Usually
Um,
the beginning of February, end of January, they'll start brooding up.
yeah. Um, and again, commenting that the drone brood is favored for varroa mite homes. Um, and you can kind of use that as an indicator of your level of infestation. That if you've got a zone of a lot of drone brood and you scratch across it, open them up, and there are no mites there, that's good news. Yeah, BIP has their scale. I don't know that it's used, maybe perhaps in some reports or printouts that the measurement of the brood uniformity is on a scale of 1 to 5, uh, where 1 is the healthiest and 5 is the worst. Visualizing cast and scale in the empty cells may require a magnifying glass, but another handy thing is a pair of strong reading glasses. Another picture of slipper-shaped sac brood, again, a virus infection. Nice, strong, uniform brood as this. And typically there's supposed to be some nectar out here, and then the pollen out in the periphery as well for the most ideal. These are tests that you can use to identify the foul brood. Costs about $15, $16, $17 a piece. If you need that confirmation, you can get that from Amazon. Now here are the spectrum of viruses. A chart like this is not appropriate to really assimilate in a quick PowerPoint, but it shows the variety of them and the nature of their signs. And the more common concern is the deformed wing virus, which is spread again by the varroa mite. We have the appearance that they're dead pupa, small discolored adult bees, shrunken crumbled wings, bees with shriveled wings, and colonies with high varroa mite loads. Just a side note too, the concept of viruses has been picked up, or was picked up a long time ago, and for decades. It was the first one, the chronic bee paralysis virus. And again, for that diagnosis, we could get by sending live bees to a diagnostic lab and keep a vigil on what its potential is on a particular hive or colony. So again, repeating the theme that all disease sources are out there all the time. It's one thing to take caution about equipment, trying to sterilize it and carrying disease, but the source of the organisms are still on and in the bees themselves. I think Randy Oliver points that out. That dearth could stress when they don't have enough protein and amino acids to generate some immunity. Yeah, particularly in the preceding couple of months, that's really when they wanted to have good protein. The fall flow, as it's termed, the fall flow of blooms, the asters and the goldenrod, providing extra protein for the winter bees. There's a term for the winter bee. Anybody refresh my memory? I can't recall that term for the winter bees. Randy has it. So it's social immunity, that they protect each other.
Dionysius.
Pardon?
Dionysius. Dionysius.
Dionysius. Okay, gotta write that down. Social immunity, they're grooming each other, themselves and each other. Undertakers are removing the dead bodies. Again, the comment here for the bib out front to see what's going on when there's any trend taking place in your hive. And the big value of propolis was being used in one of our little workshops and one of our meetings. I remember somebody was making infusions out of the propolis uh, for healthcare for humans. The propolis keeping the bacteria under control, and again, polyfloral diet to keep their proteins healthy. And just a quick comment referencing the thought again of having a little community interest, either remotely or here at my place in my lab, determine background of normals to help explain dead outs when they occur. We could do some diagnostics, and keeping records over the time that determine trends. There are clinical signs, there are production levels, food additives. I'd have a comment there that the pollen patties, as they're called, that are used, I heard the opinion that it's of negligible value for the pollen substitute, the soy protein that's often in them is questionably used. But if it's actually pollen, why, it's a different matter. The reason they do go to pollen patties is for the sugar that's also infused in the patty. Randy Oliver has the comment of combine the dinks. That is the small colonies. You put them together in September, so they have a bigger mass of bodies to keep themselves strong and warm. They're patchy brood indicators, and the side view of the frame, the comb that's got casts and little material on the sides to tell whether they've been contaminated. I've heard that 8 colonies, eight hives in the yard would tend to be the maximum. Certainly there are bee yards with many more than that, but I kind of abide by that, not more than 8. Old dark comb is an increased possibility of disease spread. And I would be concerned about orchards and vineyards with the fungicides that they use to protect their plants. Yeah, just a little bragging about our hobby that we're on the increase. It's cool to be a beekeeper, subject of conversation often in gatherings. Be pleased with what you do. It's also a fun thing to talk about at parties, your beekeeping. This is a long list of suggestions to minimize how the little sickness that's there, to keep it to a minimum. You inspect your hives every 2 to 4 weeks, depending on the season. Right now, of course, is not the season to do a thorough evaluation. The comment earlier of reducing the size, or the recommendation that George had expounded on, of dropping your box numbers per hive to 1 or 2, discourages small hive beetle and winter heat loss. That's certainly recommended. Rotate old dark comb. The solar melt kits kills most bacteria and maybe not spores, American foul brood, but the solar melters really do get hot, um, come around May when the sun's beginning to get very intense directly overhead. Wax foundation is better choice than plastic foundation, even though plastic a lot of people find is more convenient. Um, Sticky boards, of course, are helpful to monitor the possible drone or mite drop that's there. Um, and your alcohol or sugar rinse, alcohol rinse or sugar shake to determine the amount of mites in your hive. Um, Monitor your queen, replace her by the second year. If you judge, again, keeping records and you know that by comparison of one colony to another that particular queen is not performing as well, why should get another one, replace her. Feed protein during dearth and drought for stronger winter bees. Careful. um, I've heard and have been guilty myself of making awkward splits that they need to be measured appropriately, that there's enough population in the daughter hive that's going to cover the brood frames. Um, it's not just a matter of taking one box off of the top and letting them figure it out for themselves, but you can switch the frames around. I've been trending to have all of my boxes as mediums, so I can enhance that um, ability to move them around at will and keep them balanced. Okay, multifloral storage. Floral forage is a balanced health diet. Ideally, your entrances to the hive should be to the southeast, and if possible, wind protected from behind. Direct sun in the mornings. Morning sun gets the southeast entrance. Screen bottom boards and other measures for summer ventilation. I've put in the sticky board on my screen bottom boards. Insulation over the inner cover in winter and the sides as well gives a help. Here's where a cluster of my hives are kept in this. It's a 32-acre meadow, so I have a lot of multifloral sources for them. So the Buddhist beekeeper says, be ever mindful of all the joy and challenges in beekeeping. Mindful is the word. And that's it. Any latent questions here?
Yeah, Ray Walker's asked, hey Ray, good to see you. Ray's asking, what is the current best practice regarding nosema infection threshold and treatment strategy?
I think nosema is overstated. Um, that would be the topic of discussion if we have a cluster of diagnostics to review that. I don't have an answer for it. There's not a threshold. Randy Oliver points out that for one, the idea of spotting or diarrhea caused by nosema is a fallacy. The again, the threshold is very nebulous. It may be a co-pathogen to some degree because, yeah, you look at the details of where the nosema works in the gut, multiplying, and you'd think it really causes havoc in the individual body. But I would not treat I would not treat for nosema, period. I don't know if that answers the question.
Vidya has asked there microscopy resource you would suggest for pathogens, say 10x to 40x level. Viruses, of course, are PCR type assessment. So I guess Vidya is just asking uh, what microscopy resources are you using and what would you recommend?
Um, well, again, for the nosema, mostly as it's an indicator, um, and kind of fascinating to keep track of. That's at 400x. Um, the pollen studies go up to 900, um, and this is a light microscope. Um, I personally don't do any PCR at all. Um, and I think you've got to have live bees for that, and you send them into a lab, to have that done.
So no PCR in Don's garage yet?
No, I wish. Maybe
Uh,
with help we could work that out.
I doubt it. Vidge is at so oil immersion for pollen. How do you look for pollen, Don?
Yes.
Look.
Um, well, I have a large category or large catalog of identified pollen. It it's more of a toy than any diagnostic value. And I also have, and this might be of more interest, a microfilter of that we use to dilute honey and express it through this microfilter little cassette, and it collects the pollen that's in the honey, um, which could be informative for the beekeeper to talk about where his or her honey came from. And yes, that's the oil immersion.
Interesting, Don. You mentioned a bib. I've gotten into the habit over the last couple of years of taking the weed whacker and about 3 feet out from the hive, I just take it down to the dirt, like, and just keep that as bare as possible and use that, when I show up to the apiary just to look in front of the hives to see if
Uh-huh.
any of the hives have a dead bees or pupae or anything out front. And I find that easy to do and helpful.
Yeah, if you get it down to the dirt, that's probably as good as the surface as I've been putting a strip of plywood. The 8 feet will cover two, maybe three hives, if they're not too close together. Um, and that's nice and smooth. That was in the couple of pictures there. Um, but a weed whacker down to smooth dirt, that would serve, and you wouldn't need to bother with a cloth or a piece of plywood.
It works okay. You just gotta,
Uh-huh.
get it
Keep
all the way
getting.
down to the dirt.
Yeah.
Keep doing it. Um, Yeah, and a lot of people, Don, think that, when they go into a hive that's underproducing or not doing well, the first reaction is it's a queen problem, right? And I I think research has shown if you take that queen out of that hive and put it into another hive, oftentimes the queen ends up doing just fine. So really the factors of poor nutrition and disease oftentimes are what's causing that hive to be underproductive, while I think we're often taught that it needs a queen replacement. So just to have nutrition and disease on your mind all the time when you're seeing a hive that isn't doing well.
Yeah, that's good insight.
Yeah, and I, and to mention the inspectors, right? Your best resource, if you guys have questions about disease, surely take pictures and, contact Don or send them, to more senior beekeepers in the club. But oftentimes your best resource is the apiary inspector, Jeff in our Delaware and Chester County. So, that's a great resource. And I was with, and Jeff will inspect your hives every year if you're a queen and nuke producer, and I have the privilege of working with him every year. And he says one of the most common things that he sees is that the stressors is these early splits that people do, like you were saying, Don. Like, when you split a colony and leave that split in the same yard, and all the foragers end up going back to the original hive, and you have an unbalanced hive, and you have a hive that can't forage adequately, and then it's that nutritional stress. He said that's often the start of the cycle of getting a hive that's unhealthy, that stress as a beekeeper.
Yeah, that's a good point. Yeah.
Yeah, and the urge to make splits or make divides or increases, you end up, you really should take that split to another yard ideally, even though you can do it without, but I think you're stressing the colony out when you do that. And things like European foul brood, he said, is definitely a stress disease that you just, that's how it starts to take hold of a colony, or like any disease. Like you said, all these colonies have disease, and when you stress them out a little bit nutritionally or population wise, that's when the diseases start to become more of a problem. Anyway, uh, what's via USB connection? What sort of microscopes do you recommend, Don?
I have Leica,
Hmm.
um, and they're PC oriented. That is, I can um, see on my laptop what the viewer sees through the objective.
Vince Aloy is on tonight. Hi, Vince. Well, all the science focused people on tonight, Don, which is great. Vince is asking, Katie Lee at University of Michigan had date on the colony affecting the queen's productivity. I need help, Vince.
So
Oh,
you were
no.
talking,
Okay, go ahead, Vince. I'm sorry. Go ahead.
yeah, you were talking about the queen productivity depending upon the colony.
Right.
And Katie Lee published a study where she showed that moving the queen from one colony where she wasn't doing very well to
Hmm.
another colony can sometimes increase her productivity considerably. So you can look that paper up.
Thanks, Vince. So being a colony factor, not a queen factor, sometimes affecting colony productivity.
So as if she was unpopular with her subjects. The original hive didn't like her, and she went to a new population of
Yeah, when you take a queen out of a colony that's underproducing and you put her in another colony, and then all of a sudden she starts producing. So what was thought to be a queen issue was really, it may have been a queen issue, but it may have been some other factors related to how
oh
the
yeah.
health of that colony affecting the queen performance. Or maybe it was just the mismatch, right? Maybe that queen just wanted a new home. I'm not gonna lay well, or I'm not gonna lead this colony well until I find a new one. Who knows?
Didn't have a control on that experiment.
Hmm. Yeah, so I do think making it just a part of your routine inspections to look at brood. If you work with Jeff Eckel, his inspection, he doesn't care how much honey's on the hive, really. He's looking at 3 frames of brood with every inspection, and looking at open brood and looking at cap brood. And if you routinely do that with all your inspections, you can get quite good at detecting all the wonderful pictures that Don shared with us today of all the abnormalities.
Don, click the share button to unshare.
Pardon?
Are you still sharing or
Well, I was going to get something here that was interesting about milkweed. That they have a milkweed traps bees.
So, to follow up on Don's point on sort of an interest group on disease, I know Don has done this in the past, and I've been in Don's garage and looking through his scopes, and I think it's a hugely valuable service that he can provide. So if there's a group of people that are interested in getting together or taking Don up on the offer, the club would definitely support helping recruit members or pass the word along of what you're doing or provide you resources that we have to support that, Don. So, definitely something
Yeah,
that
that would be
could be
fun.
highly valuable.
Be very enjoyable.
Yes.
Here's another little story, if it's worth sharing, about milkweed. Can you can you see all those little tags? Those little brown tags? I expanded it. This is on the sticky board. There, there, there. That's from milkweed from in here. These little hooks will actually trap bees that died because of it. I don't know what part of the stamen or whatever that is in the bloom of the milkweed, but there there's some risks.
Yeah,
So
wonder
there's
what advantage to the plant is to do that to that poor honeybee.
yeah, I wonder it's not a carnivorous plant. It's not going to keep the bee to dissolve it.
Vidya saying it's an evolutionary conundrum. So Vidya don't understand. We're all in trouble. Nancy's asking, what's Don's email? I don't know if you want to put your email in the chat, Don, for
Oh,
people.
sure. It's doncoats@verizon.net. Yeah, everybody knows where the wax flakes come from, right?
That's a cool picture.
Yeah, the glands are right under the tergites. Been
Yeah, that's a
stung.
good point, Vidya. We have to get the club microscope out to the meetings again, so um, that's on my to-do list to maybe get Don and the microscope together at the Saturday meetings and do some microscopy this year for people who show up to meetings. So agreed on that one. I mean, we have
There's
so
the
many
heating.
wonderful resources in the club, and Don's clearly one of them when it comes to disease and disease recognition and microscopy and all that. So whatever we can do to help that for sure. Support that, I should say, not help it, just support it.
Here's the heat photograph of beehives. You can get a little adapter to your phone to do that.
It's his the season, right? For people to get out their thermal cameras and see if their clusters are alive. And
Yes. Yeah, the best time to do that is in the morning before the radiant heat affects the outside of the box.
yeah, so hopefully you'll get some, hopefully we can get your group back together, Don, in coming out of winter and get a few people back interested in doing some microscopy and talking more about healthy hives, right? Because there's a lot
Right.
to learn for sure.
And again, we can start with the dead out if somebody wants to have an investigation. Start with some pictures of the dead out. And
We usually do that at a club meeting in January or February. I know we have a pretty interesting plan for January, so stay tuned on that. But maybe in February we could do that a dead out again because I think it's good for everyone to learn the process of going through a hive that didn't survive and try to figure out what happened to it. It's a team effort, Don, right? So we're blessed to have a lot of wonderful resources in Chester County, and if we can all work together, great things can happen, right?
yep.
Thanks, everybody.